This is the Emerging Scientist 2022 virtual conference held at Pakistan. I am Adesike Kolawole, and on this particular work, I've worked in collaboration with other scientists, with Dr. Sibitoye from National Horticultural Research Institute, Ibadan, Nigeria, and Olayinka Abiodun from West African Center for Crop Improvement, University of Ghana. I'm presenting a poster titled Phenotypic Diversity of Eggplant Accession Collected in Nigeria based on Agronomic Traits and Nutritional Quality. I'm a plant breeder by training. I'm also a lecturer at Ladoke Akintola University of Technology, Nigeria. In collaboration with National Horticultural Research Institute, we're looking at the crop eggplants. This is because eggplants is an underutilized crop and limited research have been carried out on it, in spite of its nutritional benefits and health benefits. So we started the work on eggplants, looking at the pre-breeding activity that had been done, from which we moved to the crop improvement program. Looking at eggplant, eggplant is one of the prominent vegetable crop in the Solanaceae family. It's an economically important crop with high nutritional value and health benefits. The most prominent among the eggplant species complex is the Solanum melongenium. However, some equally important species and wide relatives, such as the Solanum ethiopicum, the Solanum macrocarpon, the Solanum guinea, the Solanum nigrum, amongst others, are a rich reservoir of useful genetic traits. But you'd agree with me that with the decline in diversity and potential of cultivars, almost all cultivars had decline in diversity. We need a robust breeding pipeline with broad genetic base that can eliminate redundancy in the development of lines with desired horticultural and nutritional traits. From the from the breeding point of view, one of the fundamental steps in breeding for enhanced agro-nutritional traits is the availability and or production of genetic variants that breeders can exploit to meet the increasing demand for high yield and high nutrition. In order to maximize the economic and nutritional health benefits associated with eggplant, there is a need to embark on its improvements. To accelerate the development of improved varieties, it is imperative to assess the extent of variability among existing genetic resources. That is why the National Horticultural Research Institute Ibadan has been saddled with the responsibility of improving horticultural crops in Nigeria. This institute holds a large germplasm collection of eggplants and other cultivars with limited information on their variability and utilization in breeding program. Therefore, the objective of the study are to assess the magnitude of variability in eggplant accession maintained at the Genetic Resources Unit of National Horticultural Research Institute, Ibadan, Nigeria, with respect to the agronomic traits, the nutritional and physicochemical properties. Also, we are to identify divergent eggplant accession with complementary traits, which may be useful as parental lines for hybridization. The materials that were used in the study include 14 genetic material. This constitutes of 13 eggplant accession and one popular farmer variety. Initially, in the beginning of this experiment, we had more than 14 accession, but those that were able to germinate and have yield at the end of the experiment were just 13 accession and one which was used as local check, planted by farmer in the environment in which this evaluation was carried out. The accessions were evaluated during the main growing season of 2020 at the teaching and research farm of Ladoke Akintola University of Technology, which is situated in the direct savanna agroecology of Nigeria. The experiment was laid out in a randomized complete block design and it was replicated 3 times. Seedlings were transplanted to the field from the nursery and the rows, inter and intra-row spacings according to how eggplants are planted in the National Horticultural Research Institute were followed. Fertilizer were applied and manual weeding was carried out as and when due. To protect the leaves from defoliating pests, plants were sprayed with permethrin, which is an insecticide, at planting and at 2 weeks interval. Data were collected on agronomic traits, nutritional traits, and physicochemical properties. In all, we had about 13 agronomic traits, 7 morphological traits, 6 nutritional traits, which are the proximate properties of eggplant, including vitamin C, and 3 physicochemical properties were measured, and data were analyzed with SAS. Analysis of variance was computed for the quantitative and the qualitative traits. Data means which shows significant variations were separated using least significant difference at 5% probability level. Correlation coefficient was computed to determine the degree of traits association. To assess the extent of relatedness among accessions, we used the cluster analysis. We found some interesting results from the table 1. In the column where we have variance, we could see that the result from ANOVA revealed highly significant variation for all of the traits that were measured. But for the purpose of this poster presentation, we only came up with selected few so as to fit into the poster presentation. Substantial variability were observed for the agronomic traits, nutritional, and physicochemical properties among the eggplant accession. When looking at the diagram, the pictures for the leaves and the fruits, we could see that there are variability in terms of leaf shapes and pattern. Some are broad, some are thin, some are extremely thin. For the fruits, we could see different colors, different size of fruit size. Some are so tiny with numerous fruits per plot. Some are oval in shape, some are round in shape. So there are diverse variability that can be exploited for improvement. Significant variation detected for some selected traits had maximum value with more than 2 folds greater than the minimum. Number of fruit per plot, the fruit weight, which are important for farmers because these 2 particular traits will determine the yield. Then looking at the proximate constituent ash and composition, we could see ash and carbohydrate composition, we could see that they had maximum value from table 1 above 9 folds greater than even the minimum. This variability indicates the possibility of improving the eggplant accession for fruit yield and nutritional quality. Moving forward, ranking the accession, we found some superior accession. Some accession were superior for fruit yield and nutritional and physicochemical properties. We identified about top 5 superior accessions, such as the Nile eggplant 0476, the Nile eggplant 0488, the Nile eggplant 0489, the Nile eggplant 0075, and the Nile eggplant 0074. This accession could serve as donor parents in eggplant breeding program, which is designed to improve nutrition and fruit yield. Looking at the Pearson correlation coefficient, when we're looking at trait combinations, we found that we had some strong positive and strong negative significant relationship between traits. This signifies their importance as secondary traits, which is sufficient as selection criteria. The eggplant accession were broadly grouped into 5 clusters from the cluster analysis. We had intercluster variability among the accession with respect to the traits that were measured. Looking at this now, we could deduce that accessions from different clusters could be used as complementary parents for crossing to obtain heterotic response with respect to fruit yield improvement, nutritional quality, and other ancillary traits. And now we conclude that there was a distinct diversity among the accession evaluated with respect to agronomic and nutritional traits. This provides each prospect for selection of parental line useful in breeding program for improved varieties of African eggplants. Some superior accession that we identified, we discovered that from different clusters using the cluster analysis could be used as parental lines for developing varieties with improved agro-nutritional value. Now, this is our eggplant pre-breeding story. We're trying to work on eggplant so as to select parents that will be used in our breeding program. Thank you for your attention. I welcome questions and comments if there are any.